I'm Michael Wall, Vice President of Science and Conservation here at the NAT. I'm also your friendly neighborhood curator of entomology, and we'd like you to introduce yourselves as well. So if you're representing a school or an organization here today, please let us know in the chat. You can add your organization's name and your pronouns to your Zoom name as well, and that would be lovely. So what are we here for? We're here for the state of biodiversity. Why the state of biodiversity? Because the state of biodiversity is important, right? The intrinsic value of biodiversity aside, right? I think biodiversity has intrinsic value. Nonetheless, let's put that to the side. And still, there's undeniable benefits of biodiversity and healthy ecosystems to human health and quality of life. So biodiversity matters to us as human beings. So why the state of biodiversity? Because we think everyone should have the knowledge to be strong advocates for the natural world and their own quality of life. And as conservation and advocates for sustainable land stewardship, the San Diego Natural History Museum recognizes and respects Indigenous peoples as traditional stewards of the land. Specifically, we recognize the Kumeyaay people whose ancestral homelands the museum currently occupies. We extend our deepest respect and gratitude to the Indigenous people who have lived on and cared for this land since time immemorial. As the original caretakers and conservationists, we honor their continued legacy of understanding, caretaking, and upholding the pillars of biodiversity. We've got an excellent lineup today, and it's thematically centered around initiatives operating on multiple scales of conservation of California's biodiversity. You might recall, or you might not, that in October of 2020, Governor Newsom issued an executive order with multiple implications on biodiversity conservation. And this included a pledge for California to join 38 countries, and that number might be up by now, to support the global effort to achieve protection for 30% of the planet by 2030. And if it all works according to plan, I'm pasting a link to where you can find out more information of that in the chat right now. So my crystal ball suggests that we're getting ready to see increases in funding for biodiversity conservation and climate change resiliency programs. And so today's speakers are really meant to inspire us to think about the world of possibilities. And our hope is that this inspires you all to start thinking about creative and inclusive ways for our society to tackle one of the greatest threats facing our planet. So with that, we'll go into the talks. And we're going to start off with Dr. Hollis Woodard. Hollis received a Ph.D. in biology in 2012 from University of Illinois, Urbana-Champaign, where she worked with Dr. Gene Robinson on the molecular basis of social evolution in bees. From 2013 to 2015, she was a USDA NIFA postdoctoral fellow working on the nutritional ecology of bumblebees with Dr. Charlene Jha at the University of Texas at Austin. She is now an assistant professor of entomology in our own backyard up at the University of California, Riverside, which delights me as an entomologist to have a kindred spirit so close by that loves bumblebees. And there she leads a diverse lab group whose research is focused on understanding and conserving bumblebees. Hollis, thank you so much for joining us today. Look forward to your talk.
Thank you so much for that introduction. Let's see, let me get my screen share going. How's that? Can folks see it? Does it look good? Okay.
Yep, that's good.
Okay, perfect. Well, as Michael mentioned, my background is more in functional genomics and molecular studies of bees, but I've become increasingly interested in trying to find ways for my research to interface with conservation research and action. So I'm honored to be a speaker in this group, and then seeing the affiliations of folks pop up in the chat. I'm honored to be speaking to this group of people too. So my background's in molecular work, but I've become really interested and focused on bumblebee conservation because unfortunately they're one of the groups of insects that we have some of the most compelling evidence for decline. One thing to tell you is that here in California, we're in a bumblebee biodiversity hotspot, really the entire Western US. There are 25 California species, that's half of all North American species and 10% of the global diversity. So we're in a special place for bumblebees, and this is driven in part by the floral diversity that exists here. So we're in a special place. We have a lot of characters here. Many of them follow the color pattern that I'm showing on the bottom left, making it tricky for people to identify them to species sometimes, but and there are other color patterns too, but this is just a kind of a standard color that you see. I want to take a second to talk about the history of bumblebee research in California and sort of tip my hat to Robin Thorpe. Robin was a professor of entomology at UC Davis, and he wrote the authoritative text on the bumblebees of California. It was published in 1983, and things have changed quite a bit since then, unfortunately, in terms of where we find species and their status. I'm going to talk about that just a tiny bit. So things have changed, but nonetheless, this is an amazing sort of overview of the status of things, at least as of the late 80s. And one of the important things that Robin did, among many, many important things for bumblebees, is he detected some of the first evidence of decline of Bombus franklini. This is a species that's found in the far northern part of the state and southern Oregon, and we're hoping that it ends up getting listed on the federal endangered species list soon. So fingers crossed for that, so that it can afford some protections for this bee. So I'm involved in a few different projects related to bumblebee conservation. One is focused on the rather unfortunately named Bombus crotchii. This is a species that I had the pleasure of seeing live just the other day for the first time. So it's pretty rare, and I was pretty excited to find it. So I'm showing you in this figure on the left, these are historical records of this bee. Until the late aughts, and the little crosses are observations, and then the green is modeled suitable habitat based on those observational data. And one thing that you can see on the left is that this is a bee that used to be found all the way up to the northern part of the state. It was found in the Central Valley. You know, it was never super relatively abundant, but you could find it places. And what I'm showing you on the right is the observational data for this bee since the late aughts. And it's been essentially lost from the Central Valley, although there are a few sporadic records. And then the southern part of its range, it's really hugging the coast a lot closer than it used to. And so this is a bee that you might have heard of was because it was proposed to be put on the state endangered species list. Unfortunately, it wasn't listed, but hopefully that effort will continue. But this makes me really sad to say that there are some really alarming patterns going on across the state that are currently unnoticed. Right? It doesn't seem like people are really paying attention. So this is Bombus vossazinskii, the yellow-faced bumblebee. It is the most common bumblebee in California. You can see it many, many, many places. It's a beautiful bee. So many people think that it's doing just fine, but if you look at here, I'm showing you again from until the late aughts on the left and from then on the right. If you look at the suitable habitat for this bee, again modeled from observational from records, it looks like this species too. There's some things going on with it that are also, I think, alarming. So we're not seeing it in the Central Valley anymore like we used to, and we're not seeing it some of the places more inland. So I've seen it here in Riverside, for example, but it's still really rare. So there are some patterns that are going on that are unfortunate, and apparently we're not really paying close enough attention to these bees. And just because something's relatively abundant doesn't mean that it's not also in trouble as well. So I just want to make that point. So research in my lab group is focused on bumblebees. We are all bumblebees all the time. So this is the only group of bee we worked on. We dabbled in centris and we've dabbled in a few other things, but I just keep coming back to my bumblebees. We work on nutrition and social biology and the nexus between those two things. And one of the areas I've become really interested in, and as one of our lab members, Rafi, is showing you here, she's caught a queen bumblebee in this file, are queen bumblebees. And bumblebees have an annual life cycle where the queens every spring, they start new nests. So a new cohort of queens will initiate nests. And at this time, when the queens first start the nest, they are essentially single moms. They're doing all the work in the nest. And it's not until their workers emerge a little bit later that the moms have help. And so this stage in the life cycle where the queens are starting to get their nests going, we think we lose a lot of colonies at this point, but we really don't know. And so in my lab research, I'm focused on trying to understand the behavior, the physiology of queens at this stage. And then we also have a project we're starting this summer in the Sierras where we're going to be looking at survivorship in meadows in the Sierra Nevada mountains and how survivorship might be be influenced by food resource availability. So we're trying to do lab and field and really heavily focus on these queen bumblebees. I just wanted to mention another project I'm working on that I'm really excited about. I'm part of a much larger initiative, so I'm really presenting this slide on behalf of Bradley Schaefer at UCLA, which is the California Conservation Genomics Consortium. This is a project. There are over 100 members, and they're sequencing the genomes of over 200 organisms across California. So not only high quality genomes for these species, but also doing a lot of resequencing too, which means you're sequencing additional genomes for species to try to understand population genomic variation. And the goal of this project, I love this, I'm prone to hyperbole, but this is really true. The goal is to build the most comprehensive genomic data set ever assembled for conservation science, and they're certainly on track to do that. We are just one project in this much broader effort, but we are sequencing genomes for Bombus sonoris, the bee that I'm showing you in this photo. This is a bee that's pretty common. Well, some years it's pretty common in San Diego, for Bombus crotchii and then Bombus vazozensky. And amazingly, speaker Amy Vandergast is going to talk all about how we use genetic data. So I got to not get that far into that for you, because I'm sure she's going to do a much better job than I could. But we're excited to start wading into that area of understanding genetic variation in these bees. And it is this important and often overlooked aspect in conservation research. And again, Amy will speak more about that. I wanted to give you a few suggestions on how you could help or participate in bumblebee conservation. One of the things I would say is that we really all need to advocate for listing under the California Endangered Species Act. So if you were following the news on this, there are some legalities of the listing process for insects that ended up stymieing this effort. But we really, these bees deserve protection. Insects deserve protection. So, hopefully if we advocate for that and advocate for change in the legislation as well, then maybe some of these amazing protections could be extended to these bees. Also, we need to advocate for legislation that tries to fight against climate change, pesticide overuse, habitat conversion and loss. Really, unfortunately, planting beautiful flowers in our gardens isn't going to be enough to save bumblebees. We need to make drastic, systemic changes to how we live if we want to try to conserve these bees and all things. And so we need to try to do that. And so I would say that's really this level is one of the most important levels for helping conserve bumblebees. If you want to actively participate in bumblebee research, there are two exciting opportunities I'll tell you about. One is the Xerces Society has a new California Bumblebee Atlas project starting this year. So this summer. And so this is an atlas project is where folks, community scientists can go out and look for bumblebees and collect information about that. And it gets put into a central repository. And these kinds of efforts help fill in some of the data gaps that we have across the state in terms of where bumblebees are found. So this project, I'm very excited about it. You can also participate in, let's say, I would call this an immersive research experience, where a former lab member, Michelle Duenas, is leading an Atlas Obscura trip. So you can tag along for 4 or 5 days and collect bumblebees with her. I'm probably going to make a guest appearance because these trips are really fun. We did one in 2019. So it's going to be in Sequoia National Forest, and they're just super fun. You get to hang out and learn about bumblebees and hang out. And it was really fun for us to kind of break down the barriers between, scientists in the lab and working out in the field with community scientists. So that's one opportunity. And then I'll just say, educating yourself about bumblebees, the authoritative guide to the bumblebees of North America is called Bumblebees of North America. So I recommend checking that out. And then I'm always happy to share other resources if you want recommendations about, queen bumblebees or any of the species that I mentioned or anything. I'm happy to share those resources with you. So with that, I want to thank everyone. I've highlighted some of the folks in my lab that have done some of the research that I mentioned or are involved in the projects I mentioned. And then I'm collaborating with Leif Richardson, Neil Williams, and John Mola on some of the work to assess the status of bumblebees in California. And then just a huge thanks to Robin Thorpe for all the amazing work that he did here in California that helps us understand how bumblebee patterns today track or don't track with those patterns more historically. So thank you all.
Thank you, Hollis. Fantastic. That was great. We are going to keep on cruising through our presentations here, and do want to throw people towards the chat. Hollis brought up that that bumblebees, there's some confusing legalities associated with listing bumblebees in California, and I wrote a blog article on that you can click through to find out a little bit more details of understanding the interesting intricacies of that. So we're going to move on. Thank you again, Hollis. Next, we've got Christa Horn. Christa serves as the excuse me, San Diego Zoo Wildlife Alliance. San Diego Zoo Wildlife Alliance as a conservation program manager in plant conservation. In this role, she works to conserve the rare plants of California through research and managing applied conservation projects. Christa earned her bachelor's and master's degree at in Earth Systems from Stanford University. She draws on her interdisciplinary background in ecology and botany and anthropology and geography to carry out her work. And with the San Diego Zoo Wildlife Alliance, she's worked on demography and sustainable harvest of Latin American palms, coastal sage scrub restoration, cactus run habitat restoration, and more. As coordinator for the California Plant Rescue Collaborative, she currently works with botanical institutions across California to coordinate our ex situ conservation efforts, especially seed banking, sharing of data, and fundraising. And she's going to be talking more about that with us today. Christa, thanks so much for taking the time to be with us.
Thank you for having me. And as Michael said, I'm talking about the California Plant Rescue. And so I'm representing all of these gardens and botanical institutions that are listed here as I go through this presentation. So as Hollis mentioned, one of the reasons we have a lot of bumblebees apparently is there's such a rich diversity of plants in California. So there are over 6,500 taxa of plants, and that is largely because we encompass a lot of the California floristic province, which is an internationally recognized biodiversity hotspot. And with that, that means there are a lot of endemic species that are only found in California. Also related to the rich diversity here is we have a lot of rare plants. There are over 1,000 taxa that are ranked by 1B by CNPS, which just means that they are rare, threatened, or endangered in California and elsewhere. So that is, at a conservation standpoint, pretty daunting that there are so many rare species in California, and what are we going to do about it? Also, when you consider that very little is known about some of these species, a lot of the 1B species, we kind of just know that they're rare, they have naturally occurring small populations or something, but there's not people regularly monitoring these often. We don't really know much about their ecology or what's threatening them sometimes. And also we're short on funding to do recovery efforts and plan those efforts. That takes a lot of time and effort. So what can we do? Well, very recently in 2019, we got support to enact part of the California Biodiversity Initiative, which was set out in September of 2018. And we got $3 million to expand seed banking and collections to hedge the bet against extinction, with California Plant Rescue, our group, focusing on making these collections and serving kind of as a state seed bank, even though we're a dispersed network of seed banks. And this funding is really helping us drive us towards our goal, and we're making great progress. So yay, that's great news. But maybe you're wondering, why seed banking? Why are we putting all this money towards that? So seed banking is a recognized conservation tool for plants that is used across the world and is really an important safeguard for biodiversity. We collect seed, we dry it down and freeze it for long-term storage, and it really is great at conserving the genetics of a population. Right here, you see a technician from Santa Barbara Botanic Garden with a bag full of seed, and each of those seeds is an individual. And there's been research done that and all those individuals are from one plant, and that is the mother plant, the maternal line. And so all those individuals in that bag are siblings, or half siblings, probably technically. If you collect from 50 mother plants, you have a really good high probability of getting 90-something% of the genetics of that population if you are randomly collecting across that population and getting good dispersal. So it's a very good way to conserve the diversity of an entire population. Versus living collections, or even the loss that might be being experienced in the wild, you're holding on to that diversity without there being selection in a garden as you're growing plants out, and maybe some things survive better in propagated scenarios than others. And you're not being as subject to drift. There is some loss of diversity possibly within storage, but not as bad as in other scenarios with cross generations. Also, seed collections are not intended to just be storage away in a bank. We do make withdrawals from our banks. They're intended to support the species survival and reduce the risk of extinction of the wild populations. And it does this by providing plant material, either seeds that are bulked and grown from the ones that we collected, or the ones we collected directly, or we're able to grow plants out for planting. And then also it provides material, those plants or seeds, for research, so that we can learn more about these species and what it will take to actually bring them back. It is also a really straightforward process. Might take hours and hours of cleaning seeds, and may take you a long time to get to field sites, but you do just collect seed, dry it down, and freeze it at its essence. And so that makes it pretty cost effective. The Center for Plant Conservation did some work that the average seed collection was between $2,500 and $3,000 to make and conserve long-term. Okay, so that's seed banking and why it's awesome. Who is California Plant Rescue? We are a group of botanical institutions across California who, after a paper done in 2014 that looked at seed banking across the state, saw that there was a real need to focus on these rare species. We weren't doing a good job of those rare, threatened, and endangered in California, unless they happen to be listed by the federal government or the state government as endangered. And that's because there's funding associated with those listings. And so we realized that we needed to come together and be a little bit more proactive in our tactics. Some of the institutions within CAPER have seed banks. We have 6 established seed banks, but we are 11 institutions, but everyone involved has botanical expertise. And we've come together to share our lessons learned, our progress, do fundraising together, share protocols, be on the same page. But our main accomplishment has been, switching to goals. Our main goal is actually to represent all of California's diversity within seed banks, which is at 6,500 species. It's actually more because we want to spread into the California floristic province and do Baja as well. So it's very ambitious. This group will be together for a long, long time. But we realize that the rare species are the ones that need the most help. So taking the global strategy for plant conservation, which came together under the Convention for Biological Diversity, their goal is to get 75% of threatened plant species in collections, and with at least 20% of that available for recovery and restoration programs. So that's kind of been our guide, with 1 bee species being our main target. And as of 2019, there were 1,166 of those. So that's what we're going after. As I tried to skip ahead, sharing data has really been one of our big accomplishments. And this has really let us know not only what we've already collected and what needs to get done and track our progress, but we've been able to do gap analyses and see areas where we should target. So one of the things we've seen is that national forests are really well botanized because they do a lot of work out there. And they have a lot of rare plants that could use collection. So we've used this as a guide to kind of help us with our permitting and who are seeking out our partners. The data from our collections is also paired with information from the Fish and Wildlife, the State Fish and Wildlife and the Natural Diversity Database. So we're able to see these gaps. We've noticed that the northeastern part of the state is really poorly represented within seed banks. And so we have plans to go out there and do a big expedition together. And we're also set up so that we can better communicate our goals and know who's working on what species within this database as well. So in marking our progress, before CAPER, some of the institutions have been baking for decades. And so there was a fair amount, around 30% collected before we started. We've been chipping away, making 50 new species collections a year. They do a lot more collecting in general, but often those species are already represented in a different collection. And so we're about 50% in 2019. And that's when CNPS led an effort with all of us supporting to seek the funding to really be the kickoff to that biodiversity roadmap plan. That this was a really achievable goal to bank the rare species in California, and a really important step for all those reasons I discussed earlier to help prevent plant extinction within California. We had the existing infrastructure with our different institutions. We had already been partnering for a while. We were ready to take this on. And so they thought that $3 million would be an effective investment to get all those 1B taxa over 5 years. In 2020, we made great progress despite having a pandemic and lots of wildfires limiting access to national forests in particular. So in 2019, we have a lot of institutions in the South. And so a lot of our collections had been focused there. We have a few in the Bay Area. You really see that in 2019. But in 2020, with that funding, we felt secure in going a little further afield. And we have much better representation across the state. It was also a record amount of collecting. We were chugging around on about 50 species a year additions. And you see that slope suddenly peak up, and we got almost triple that in 2020, despite all the challenges we had. So our goals are very achievable, and we have about 420 species or taxa left to collect to meet our goal. So very achievable. We didn't just put the money towards the collections, although that is obviously a huge part of what we're doing. But we really needed to make sure we were actually securing these collections. And so we did things like invest in backup power for the existing seed banks. Curation is very important. So making sure that those seeds are actually alive. And if they're not, if they're starting to die off, then we need to recollect or grow them out and get the seed from the ones we're growing out. And so germination chambers to test seed quality were added to 3 of our institutions as well. And we're also thinking about the future and the other things that our group is going to be doing. We're thinking about ways to conserve species that can't be banked by conventional means because they can't be dried out. These are things like oaks with very recalcitrant seeds. So we have cryopreservation, which is putting tissue or seeds in liquid nitrogen for long-term storage. And that's an option for species that are very large and maybe aren't going to be grown within a garden with the hundreds of individuals to maintain diversity. And we need to recollect from some of these populations that just have very low seed production. And just forging better partnerships with land managers and making sure that we're meeting their goals as well and what they need for their plant conservation needs. And please learn more about all of the awesome conservation work that these plant organizations are doing in California and in other places as well.
Thanks so much, Christa. That's incredible. It's really, I think, you know, $3 million is such a drop in the bucket in the big picture of investment in conservation within the state of California, and what you guys have done with it is incredible. And the goals are, you know, it just totally makes so much sense. So thanks for taking the time to present that. And I know we had several questions in the chat, and we'll come back around to those at the end after everyone else has had an opportunity to talk about their work. We're going to move on now to Dr. Amy Vandergast. Amy leads a research program in conservation and landscape genetics and genomics at the USGS Western Ecological Research Center at the San Diego Field Station. Broadly, her research focuses on understanding how natural and human-induced landscape and environmental change impact populations of native species and shape evolutionary potential. She characterizes population dynamics through surveys of genomes in many individuals and many populations across species ranges, and that's what we refer to as population genomics, and works with local, state, and federal management agencies to apply genomics to the protection of rare and endangered species that include birds, plants, herpetofauna, and invertebrates. Amy, thanks for being with us. Looking forward to it.
Thanks so much, Mike. It's great to be here and be a part of this great panel. I'm so excited to hear about all the great work that Hollis is working on with bumblebees, and the work that Christa shared with us is really exciting as well. I am going to just speak very broadly about why genetic diversity is a fundamental component of biodiversity and how genetic metrics can be used as indicators in our conservation roadmaps. Can everybody see my slides? I forgot to ask. Yeah, okay, great. Yep. Okay, so just very briefly, what is genetic diversity? We consider genetic diversity to be a foundational component of biodiversity, and that's because it provides the raw material for evolutionary change, and thus it underlies all other levels of diversity and complexity that we see in natural systems, populations, species, and ecosystems. And it also underlies adaptation to changing climate, habitat, and biotic interactions, which we're seeing are very important because change is very rapid right now in the Anthropocene. So again, very, very simply, the source of genetic diversity is mutation. And then recombination acts to kind of mix up the genome content within individuals during reproduction. And then in a population, genetic diversity is sort of further affected by the population size, how many individuals are in a population's connectivity to other populations for movement and gene flow, and then also the process of selection. And so when we want to survey genetic diversity across populations, we want to look at many genes, so a genomic approach, many individuals, and many populations within a species to determine how that diversity is distributed. And we typically focus on three metrics: the diversity of gene variants within individuals, that's the heterozygosity, the overall number of gene variants in a population or a species, we call that the allelic richness. And then also we can look at the number of breeding individuals that contribute to that diversity, and we call that the effective population size. And that's an interesting metric. It can be measured both with genetic data and then also estimated really well with demographic data. So we can monitor these components of diversity over time, along with how genetically distinctive different populations are. And what we see is in many cases that diversity decreases and the distinctiveness among populations increases with urbanization and other disturbances that decrease basically habitat patch size and connectivity. So this is one example. This is the endemic Coachella Valley fringe-toed lizard. And it specializes on open dune habitat, much of which has been lost in the Palm Desert, Palm Springs area where it's endemic. due to urban development. And so what we found is a strong signal of increasing divergence among patches over time and a significant loss of allelic richness in 4 out of 7 of the monitored populations. And this is just one example. We've seen a similar pattern in several other systems that we have done genetic monitoring over time in, including the San Francisco garter snakes, the Mojave Desert tortoises, and then also coastal cactus wrens in Southern California. So unfortunately, this pattern appears to be really common across different areas and species. So here's an example from a paper that looked across 41 different data sets for North American mammals, and they found similar patterns to what I just showed you. So for these different correlates with urbanization, whether an area was more urbanized versus rural, the level of human population density, and this human footprint index, these were all correlated with reductions in these measures of diversity, effective population size, gene diversity, and allelic richness, and correlated with increases in population structure, here measured as FST. Another paper that looked at approximately 100 studies globally found that on average, there's been potentially a 6% loss in genetic diversity across all species. So these trends are worrisome because loss of diversity often predates and contributes to species extinctions as part of this extinction vortex process. So what happens is if we have a large population that becomes a small population, it's subjected to more inbreeding, that's breeding between close relatives, and random loss of genetic diversity. So this loss of genetic variability then potentially reduces individual fitness and the adaptability of the population, which can then lead to lower reproduction and higher mortality and an even smaller population and so on. So if we're starting to measure this loss of genetic variability through time, this can be a trigger for conservation action. And we see genetic diversity as a fundamental component of many conservation plans that we have, both at the state level, also at the federal level, and even at the global level. So for example, in the California State Wildlife Action Plan, many of the fundamental goals, including these two, do focus on genetic diversity, wanting to sustain and enhance it, as well as sustaining and improving connectivity for genetic permeability. And nationally, we see this as well in the new U.S. Fish and Wildlife Endangered Species Act Species Status Assessment Framework. This is a way that the U.S. Fish and Wildlife Service is helping to adopt a more quantitative approach to basically monitoring species for conservation action, either listing through the Endangered Species Act or other actions for listed species. And it is basically using these 3 levels, the 3 R's of conservation science: representation, redundancy, and resiliency. And genetic components are part of each of these, with representation including the breadth of environmental and genetic diversity measured within and among populations. Redundancy can be assessed through the number of either genetically identified or otherwise identified populations and the degree of connectivity among them. And then resiliency really focuses on this genetic effective population size and the adaptive capacity. And then we can scale all the way up to the global level with the UN Convention on Biodiversity, which is increasingly using genetic diversity in its targets. So in the 2011 to 20 targets, there was one target to conserve gene pools here, and it was mostly focused on plants and animals that contribute to human systems, mostly cultivated and farmed, and wanting to maintain this diversity. But in the post-2020 strategy, at least in the draft, it looks like genetic diversity is now going to have a more prominent position as one of the three main components of biodiversity. And then hopefully more targets and metrics will follow. So this begs a question, although many of these plans from local to global levels, they're calling for the conservation of genetic diversity, we also need to think a little bit about what are actionable targets for these metrics, and what metrics can we use to measure success. And I think these metrics need to be relatively straightforward and simple, and also quantitative. So some have been proposed for the global level here. So an overall goal, which is pretty ambitious, halting the loss of genetic diversity within all species by 2030. And then by 2050, making sure that evolutionary adaptive potential and long-term maintenance of genetic diversity are coming on board for species that have been restored and secured. So the indicators are kind of what I'm interested in here that they've used. So the indicators here in orange, they have three that they've proposed, this group has proposed. The number of populations within species with an effective population size above 500 compared to the number below 500, as well as the proportion of populations that are maintained within a species, and then also the number of species and populations in which genetic diversity is being monitored using DNA-based methods. So these are starting to really put focus on genetic indicators for the health of populations. The focus on any, I think, is an interesting one and a good one. There's some good reasons to look at this effective population size as an indicator, because it can be assessed both with demographic and genetic data. So it doesn't fully rely on just genetic data, which is still coming on board for many species. And also there's good theoretical and empirical data that suggests that there are some good thresholds to use that can help us figure out the health of a population. This is the 5,500 rule, with a minimum of 50 breeding individuals to avoid inbreeding depression, and a minimum of 500 individuals to retain this evolutionary potential in the long term. So I want to bring it back locally now and talk about how in San Diego we're using genetic metrics along with others to assess the health of our preserve system. So SANDAG, San Diego Monitoring and Management Program, has developed or is in development of these different metrics, which they call the State of the Preserve metrics. And so as an example, I wanted to show how genetic effective population size is being used as a metric to evaluate mountain lions. So it's one of several metrics, but it's the first, is the effective population size. So based on really comprehensive genomic and genetic data that's been gathered for mountain lion populations in the Southern California area, they've come up with a target for San Diego of having an effective population size of 30 breeding individuals in San Diego. And while that might seem low compared to those targets, we have to remember that mountain lions are very wide-ranging species. Each individual needs a lot of area. And actually, San Diego is connected to other areas. So it's only a part of the metapopulation. So the total metapopulation, which would include Orange County and San Diego, then would have an effective size of 50 as a target. So based on the genomic data, the current effective population size in San Diego County is estimated at 31.6. So then this gets folded into this conservation, whoa, my screen just went blank. Okay, conservation dashboard that's being produced for all of the species and systems that are being preserved in San Diego. So for the mountain lion, the condition here is considered good for this metric because it meets the target size of 30 individuals. The confidence in this estimate is high because it's based on an abundance of data and research. And the trend still requires more information. So, some sampling further in time will hopefully establish whether this trend is moving upwards, staying stable, or hopefully not moving down. And then as my last send off here, I just wanted to also mention that it's not all gloom and doom. There is hope for the future. And it is possible to reverse these trends and loss of diversity with appropriate conservation action. So this is an example from Scandinavia. This is the Arctic fox, very cute, especially in the winter. So what they did in this area is they released captively bred foxes to boost local population sizes, and then did some more conservation action in these areas, including removing red foxes and then supplemental feedings. And what this did was boosted the local population size, which increased the NE, the genetic variation within populations. And then also it allowed these animals to colonize this intervening habitat area between these populations and restore the stepping stone gene flow that used to be in the system, which then further increased the overall genetic diversity in the system. So these are the types of outcomes we hope to achieve with conservation. And I will leave it at that.
Great. Thank you so much, Amy. It's you really connected our two previous talks to how that can understanding that genetic data and why it's so important to have it span, distance and individuals and distribution, all these sorts of things to better manage for the future. So thanks so much for that. We're going to move on now. We'll have time for questions for the entire group at the end. And we are going to have our final panelist, Dr. Amber Pairis. Amber is Executive Director and founder of the Climate Science Alliance with affiliations with the Southwest Climate Adaptation Science Center and the Western Regional Climate Center at the Desert Research Institute. In 2019, Amber was recognized as San Diego's Community Hero for Climate Change by KPBS and the National Conflict Resolution Center. In 2017, she was honored with the National Climate Adaptation Leadership Award for Natural Resources. In 2013, Amber was appointed by the Governor Brown as Assistant Secretary for Climate Change, California Natural Resources Agency. But for her, the real heartfelt work began in 2015 when she launched Climate Science Alliance, working collaboratively to create a space where organizations, agencies, communities, and individuals could work cooperatively to advance climate resilience on a regional scale, on top of all of the awards that Amber has been recognized with in the past. I would submit one more humble award that she's one of my personal heroes too. So thank you, Amber, for being with us today, and we look forward to your talk.
Thank you so much, Michael. Can I just take you with me everywhere? I think you should talk to my children about these accolades. Maybe they'll see some of them. But no, I really appreciate this opportunity to join this wonderful panel and to be part of this. So thank you for the invitation. I also want to start off just by acknowledging and thanking the Kumeyaay people for allowing me to live, work, and raise my children on their ancestral homelands in what is known now for us as San Diego. And I also just extend my deepest gratitude to all of my Indigenous colleagues, partners, friends for their support, their friendship, their teachings, and really just the good work that we're doing together with partners across the region, tribal and non-tribal, and breaking down barriers and moving the work forward. And that's really what this project is about that I want to share with you. I would also be remiss in not extending my gratitude to my team. I have a really amazing team at the Climate Science Alliance of incredibly committed individuals who bring their heart into the work every day. And we all carry a very heavy load together, but because it's carried together, it's doable. And so many of them are on this call and deserve to have a little bit of a gratitude clap. So I wanted to acknowledge that. But as I kind of jump into sharing this Resilient Restoration project, I wanted to give you just a little bit of framing and background on the Climate Science Alliance, because our model is really reflective in all the work that we do, and particularly this project. So if you're not familiar with the Alliance, as Michael said, this was my dream to create this space just about 6 years ago now, a space where we could come together as researchers, as managers, as foundations, as NGOs, as artists, as educators. It was really dreamed to be a space that we could look at where we could support and promote each other, and then knit those actions together in a way that they have more impact together than they do alone. And the Alliance has grown over those 6 years to now include over 360 diverse partnering organizations and agencies. I'm trying to be cognizant of my good friend Fernanda, who's doing mad translation on the other end. This model of how we do this work is also reflective in every single one of our projects. We focus on these 3 core areas. They're focused on what we call building resilience, building capacity, and building community. And the most important part of these is, particularly with building resilience, it's that translation, that dissemination of information, and then also really creating the relationships with the people who are using that information. So when you have a really hard decision to make, you can really trust the person across the table from you in helping guide what those decisions are. Building capacity is about meeting people where they're at. So whether you're a stormwater engineer or you teach 12th grade science, we really want to help you understand what climate change means so we can mainstream these concepts and make them really become part of the lexicon of everything that we do. And then building community is really probably the heart of a lot of this. It's, what are those unique opportunities we have to really work with our communities, to elevate understanding of climate science and solutions, and to really inspire and bring hope to something that's often a very sad and dismal topic to work on. And we have really wonderful initiatives with working with artists, and then also our Climate Kids program, which is really focused on supporting and nurturing our youth to be the leaders that they are now and into the future. And so that being said, the real beauty of those 3 pieces is what happens when they come together. And that's really how we approach every single one of our projects, has that component in it. And it's very focused on the community or the partners that we're working with. Our role in the Alliance is to support our partners, to facilitate conversations, to elevate their voices, to really help build that capacity. And we're successful when you don't need us anymore. That's our ultimate goal. I don't know how that fits in the bigger business model of how the world works, but that's my model, is that we're not needed anymore. And so this is, extended to all the communities we work with, but something that we're really building out and with our tribal partnerships is really around sort of changing this paradigm of how conservation work is done, of how climate adaptation work is done, and breaking out of these sectors and silos that are so easy to get pulled into. And so it's, it's really easy to kind of dig in and just focus on what you know and the people that you're used to working with. It's really easy to become territorial of your work. It's really easy to become competitive with each other over really limited amounts of funding. But we cannot face the challenges that we have with climate change if we do not figure out how to break down those barriers and really figure out how we do the work together. And that's really at the heart of this particular project with our Resilient Restoration. So our tribal partners have been, leaders have been a part of the Alliance since it began, but in 2017, there was real impetus to build a separate tribal working group. And this was a place to really focus on intertribal priorities, places where the tribes wanted to come together to work together, but also a place to bring in those non-tribal partners and really guide the work. And we've seen as the growth of this is that, this particular project and the ones leading up to it have been really coming from the advice and vision of our tribal partners. And again, this sort of goal with all of our community work, but with the tribal work as well, is that we really want to be supporting projects that are visioned by, led by, and implemented by the community. And so these, when we think about sort of long-term implementation and efficacy, if these projects are not embedded in what the people who live there and work there and raise their families there, they are the ones who will caretake this work into the future. They are the ones who should decide what their community looks like, how they want it to look, and you know, really how we build what we're always working towards in resilience and adaptation and climate change. So with our particular resilient restoration project, there's all these different pieces. There's these different players of really amazing knowledge holders. But what I really want to draw your attention to is the center of that, the heartbeat of all of this is the tribal work group. This is where all the information gets funneled. This is where the reciprocity, the relationships, the conversations, the decisions are all really funneled through that group and really embedded in this equal valuation of ways of knowing. And again, just really a strong focus on these reciprocal relationships. And I'll kind of come back to these pieces, but I just want to focus on that. We have a really phenomenal team involved in this, partners from UC Riverside and San Diego State, in addition to our Climate Science Alliance team. All these folks, I think almost everybody is here on the phone today, and they're going to be part of the breakout group. So I really encourage you to join our breakout group and meet some of these amazing people. It takes really special researchers, and there's so many out there, but really special people who are willing to sort of open their hearts and think about how they can help bring in traditional ways of also traditional knowledge, place-based knowledge, and really embed that into their practices and their own expertise. And that's what makes it such a strong team. So the real vision behind this Resilient Restoration Project really came about from just the priorities the tribal work group was working on, where they wanted to see this kind of better understanding of what climate change meant to these culturally significant plants. And I would say, you know, all plants are culturally significant. So it's really these, what does climate change really mean? And for some of these ecological systems that we focus on, but then really bridging that gap to what it means for their community. And so there's kind of these different pieces of it, of where the research is really going to be helping to make decisions and really sort of investing in the communities as we move towards the future. So again, kind of these different pieces, I'm just going to touch on the different roles and responsibilities of each of our team members. And again, how that is really integrated into the guidance and advice from the tribal work group and making you know, decisions and really leading the project. But again, just I wanted to also emphasize that while everything goes in and out of this tribal work group, there is space in that tribal work group for our non-tribal partners. This is not just about the work that happens on reservation lands. Every land you stand on is someone's ancestral homeland. So every planner, every landowner, everyone who does work or cares about biodiversity really needs to be working with our tribes, being good partners all together. And so that's where the space in the tribal work group also brings in others, so that we're really, again, sort of breaking down these boundaries. So I've sort of spoke quite a bit of this, but the tribal work group, really their role is really to lead this and guide this and to provide the expertise and to really sort of work with the researchers together as friends and partners. This work builds off of long-standing relationships we've had with Dr. Megan Jennings at San Diego State. Again, another project. These are sort of these stepping stones we've been working on together for years with the tribal work group. And this Connecting Wildlands and Communities project, which is just starting to wrap up, really brings in this kind of holistic approach to understanding really biodiversity conservation in the face of climate change, habitat connectivity, wildlife movement, and then brings that in together with water management and fire management, because we don't work in these isolated sectors. It's all really embedded in this. Again, it was really driven by you know, input from our tribal partners and continues to be sort of led and informed by everyone, and really focused also on those planners. So building off of this work as a platform, we have these amazing researchers at UC Riverside, their superpowers in modeling, and really being able to leverage a huge project they have to look at climate change impacts in plant communities, but their desire and willingness to come in and really focus on a suite of species that were the priorities for our tribal partners. And so they are going to be able to really take this robust modeling, this understanding of population dynamics, this forecasting, this climate information, and really help us understand that future suitable habitat for these suites of plant communities that, again, sort of been identified by our tribal work group. And then taking this kind of larger scale modeling and understanding what that trend looks like, understanding that vision, we then have really amazing researchers at San Diego State, Dr. Lluvia Flores Renteria, I'm hoping I got that one right, Lluvia, who's really been able to sort of hone in and help us get some traction around what's really going on with some of these key plants and communities right now. And in particular, there's a suite of plants that we're looking at around a suite of oaks, but I sort of put this picture up of coastal live oaks and really sort of breaking down this understanding. It's not just the ecological values, but it's the ecological and cultural values together. And so we know that we have these current threats to oaks with our existing stressors of development, our land use decisions, fragmentation, soil compaction, and then all this gets overlaid with these climate impacts, this drought, extreme drought, heat, precipitation variability. We have more pests and pathogens. There's all of these compounding factors. And so with some of the work of Lluvia really digging into this is helping us really understand where are some of our populations right now really resilient, some of these greenhouse experiments that can really help us detect drought tolerant families. And so this gives a picture. We'll have this vision from our UCR colleagues, Janet Franklin and Helen Reagan, about what the future vision looks like. Then we have this information of what things look like right now for some of these plant communities. You know, where are things really kind of on the edges of their range right now? And what will that mean for the future? Where are things in their sweet spot? So they can really help us understand how we, you know, where we invest in time and energy and how to conserve and protect those things. And I will say that even if they're on the edges, that doesn't mean that we're leaving anything behind. I'm going to get a t-shirt made up that says no oak left behind, because these are not just plants. They're not just trees, they're relatives, and they're an intimate part of the whole system and this larger stewardship component that we're really supporting at the behest of our tribal partners. So then all of that comes together, right? All of this understanding, this knowledge holding that, you know, whether it's the knowledge that our tribal practitioners and partners are bringing, whether it's the knowledge that people who are working on the ground every day bring, whether it's the modeling, the research, the crunching of the numbers, to then really look at how you integrate all that into real world shovel-ready projects. You know, climate change is this global phenomenon, but its impacts and the application of strategies and actions are inherently local. They're place-based. And so we'll be able to take this information and really work together to develop strategies, to really prioritize actions. San Diego State has offered to be part of this in terms of developing a business plan. If there's ways that we can build capacity in the communities to carry the work forward, what does that look like? What does it entail? And then really also being able to make sure that data is accessible, available, and in formats that people can use it. And I don't have time to really talk about data sovereignty, but if you have not heard that or know about that, please, you know, educate yourself on data sovereignty, because this is a really important aspect of being able to protect information that's sensitive in a way that honors the people that we are working with, and being able to make sure that their data is available. And then all of this sort of will manifest into pilot projects, into testing strategies, and to really trying to get work going on the ground that's reflective of the amazing amount of thought and research and energy that's gone into it. So I just want to sort of wrap up and close the presentation with just this, you know, sort of reiteration of the importance and value of relationships. Relationships, reciprocity, and respect. Again, you know, we have so much ahead of us, and if we don't figure out how to break down these silos and work together, then we just won't be successful moving forward. It won't matter how good our research is or how much money we pour into something if we don't figure out how to do it together and do it together the right way. There's a really amazing book, if you haven't read, by Frederick Joseph called The Black Friend. And it's an amazing read. It's very accessible, it's very personal. And he talks a lot about that it's not good enough to be an ally. You have to be an accomplice. And I feel like the word accomplice, you know, it has this sort of negative connotation of, you know, an accomplice to a crime. But really, when you think about the word accomplice, it really means that you have to be completely committed. So it's not enough to empathize that something is wrong. you have to do something about it. It's not enough to recognize that you need more seats at the table. You have to not only make the space for those seats, but you have to step back and allow space for others to step into those leadership roles. And I think that this project in particular, as I hope sort of to communicate, is that it's through that, those relationships and making space that will really create the equitable and sustainable and resilient future that we want to see. And I think I've listed some things in here of what we're trying to do really specifically with this project, but I just would like to end it with just a short story. And that story is that, as many of you know, when you get these grants, like it friggin' takes forever to get everything signed and the administration and the official like start date. And so we were a little bit behind in what the timeline that we thought. But as things like, kind of came together, we realized that we had sort of running out of time in getting these greenhouse experiments going. And that we wanted, Yuvia and Megan really wanted to make sure that we got things going so that if things didn't work out this first year, that we could fix it and then have a really robust experiment in year 2. And so under other circumstances in different groups, those researchers, they could have just gone out and gone collecting acorns. They could have gotten the permits that they wanted from federal and state, land managers and just gone out and done it. But instead, they went to the tribal work group and the relationships had been established. We were having conversations about this project and they were like, we're running against this timeline. We really need to gather acorns. How can we do this? And our tribal partners stepped up. They dropped a lot of things they had to do. They figured out how to either get access for Yuvia and Megan to go collect, or they went and collected for them, or they went out there with them to make sure that they could get the acorns they needed and start that propagation. And so the goal was always that any acorns that were part of the experiment would go back to the community. Well, with all the acorns, Yuvia called us up and she said, the babies are sprouting, and like, there's so many acorns and, I have enough for the experiment and there's all these other acorns that are sprouted. And, what can we do? We need to get them back. And so this is, Megan collecting everything. We divided everything in our cars. We drove everything back. I swear I might have driven more carefully with those acorns in my car than I normally do. I felt like I had precious cargo, but it is, right? And it's about respecting, that relationship with our tribal partners, that these acorns, these seedlings are important. And so those seedlings are now home. They are in pots, they are growing they were given back to the tribe. And that's really what I hope, gives me hope, because it's about the reciprocity, the relationships, and the respect. And I hope that that inspires you to be an accomplice and, to really bring this into your work. And thank you for the time to share that with you. And I hope that you will join the rest of the team in the breakout sessions with us after this. Whoo, Michael, did I do it 10 minutes? I think I went over.
Yeah, sure.
Thank you. And thank you, Fernanda. I know I talked fast.
Thanks, Amber. And I think it's interesting. We sort of had bookends, right? Hollis talked to us about a very specific group of organism, bumblebees. And one of the things that she said in her presentation was, it's going to take systemic change to conserve bumblebees. And here at the other end of our bookend, we have Amber talking about this really integrative approach that is systemic change. It's an effort to bring about systemic change. And so I hope you've walked along with us on this journey to look at the way that we can think about conservation of biodiversity in Southern California, and enjoyed it. We normally would take a 5-minute little bio break here, but we're going to take a 3-minute bio break. So you got to be really quick. Okay. So, but we will return back here in 3 minutes, which means 10:16, according to my clock. That's odd enough, but we'll go for it. And we'll see you back here in 3. Thanks, gang. Excellent all. I want to again thank our panelists for being with us, contributing their time and talent. I found all their work to be inspiring personally. I also know many of these people, or at least am friends with the folks that they work with, and they're all incredibly passionate human beings, and I hope that you saw that passion as much as I did in the presentations today. I was looking through the Q&A in the chat, and several of the questions got answered in the chat itself. And then there are a few that have not necessarily, but in the interest of time, I think what I'm going to do, because I think that these breakout groups are going to be hopping. I think there's going to be a lot of interesting potential for discussion and activity in the breakout groups. We'll talk more about that in just a second. But instead of going into the questions that people had in the chat, I would encourage you to save those questions and take them to the breakout groups with you. Instead, I want to talk to the panelists about hope. And so Amber finished her presentation with what makes her hopeful. And you know, if you joined us in our very first plenary speaker, we had Nancy Knowlton speaking to us about Earth optimism and the resiliency of ecosystems and species. And so I'd like to open it up to the panel, and we'll just go in the order of presentation. So Hollis, we're going to start with you. You know, what are the things that make you hopeful about bumblebee conservation here in California?
Well, I'm sort of inherently optimistic, so I can't help but be optimistic. But I think, you know, I mentioned that these big structural changes need to happen. I think some of the movement on climate that's happened in the new administration, and some of the social justice issues that have happened recently, I think all of those things are part of a much bigger fabric that's ultimately going to be the change that we need to try to conserve not only bumblebees, but all living things. So yeah, I think I see the good in things and inherently, but I also really do see some positive things happening, especially lately, that make me think that the fundamental shift in how we think about things and how we treat people and how we treat our ecosystems, that those things can and hopefully will happen.
Great, thanks, Hollis. And there were some specific questions for you in the chat, so I'm hoping that the people who asked those questions will join us in the breakout group, because and I'll be in your breakout group as well, because I can't hesitate to take the opportunity to talk bumblebees. So let's how about you, Christa? Where's the hope in plant conservation here in California?
Yeah, I think just the amazing things that you can do as like collaboratively with a group, I think is really interesting. So my talk obviously focused on that, but like just as an example of a withdrawal made from a seed bank at Santa Barbara Botanic Gardens, they have a very ambitious project on the Channel Islands with many of the rare species there, and they're taking seed that was collected from over 20 years ago and working with some propagating that out in a restoration, and that involves federal partners and Nature Conservancy, all these different elements together, coming together and making progress on saving some of these species. So all these groups coming together and able to accomplish a lot. It's amazing, and we saw that in Amber's talk as well. Like alliances are key.
Yeah, excellent. And Amy?
I've had just a really, I feel like such an amazing opportunity to get to work with so many different people and different agencies and conservation groups and my own lab and other labs. And I think what brings me the most hope is just the amazing amount of commitment and drive and ability to think outside the box, and everyone just working so hard. And it's hard not to be hopeful when I see so many brilliant people every day just coming up with these great ideas and putting them into action, and everyone working together. So in that sense, it's very hopeful. I also think it's just being a geneticist, a very exciting time to be in genetics. There's so much happening in terms of our ability to really get a more genomic picture of what's going on, look at genes under selection. There's so many new avenues opening up. And so I'm very hopeful that as our science becomes better, that we can then apply that in better ways for conservation.
Excellent, thanks. And Amber, finish this off.
Oh, I think it's just the power. I inherently believe in people, and I think it's the power of us all coming together that is really where we'll see that. And I think, you know, seeing that in our youth programs, and also these sort of multi-generational dialogues that include, you know, our elders, our grandparents, all of these knowledge holders, no matter what that stands for. We have a lot of good things coming. I think we feel it, right? I think there's a change in the air, and I think we're on to something. And I think we just have to strive to figure out how we bring those lines together, because this is not the time to be competing with each other or be territorial. We've got to figure out how we do this together.
Totally, absolutely, I couldn't agree more. Well, I've said it, I think 2 times. I'll say it a 3rd time for our 3rd session, and that certainly what gives me hope is hearing folks like you passionately talk about your work and the talent that you bring to the conservation of biodiversity within our region, and the inspiration that you give to other people to believe that we can do this. So thank you all for joining us today. It's always a pleasure to share a screen with people as talented as you.